Hello and welcome to another practice session for IIT JAM 2022. This is basically from microbiology. This is practice session number 3. Other practice sessions and previous year questions are found in the I button, so don't forget to check it out. In this video, we'll practice 10 MCQ questions based on previous year questions and the most probable types for IIT JAM in 20 minutes. But before that, let me tell you about if you are looking for quick tips and tricks for the last moment, you should consider joining Unacademy Summit Batch, which is a 30-day question bank related courses for Unacademy. Here, the educators would provide you with question banks based on the previous analytics and also the most important type of questions that are asked in IIT JAM. And it's starting from 19th January, that means day after tomorrow. So overall, you should get high benefit from these particular batch because it's high yield topic. You can use my code AP10 to get a 10% discount, and don't forget to register yourself right now because Unacademy's educators work a hell lot to get all of these informations gathered in one place, compiled nicely for you, and it would help your preparation. So don't forget to register for this particular course. Now you might be thinking of the financial aspect. 1 month subscription for Unacademy really costs nothing. Don't think of like small term investment or small term subscription. Think about the long term. If you want to give IIT JAM down the time 2 years, so a 12 months or a 24 month subscription could be very useful for you, and for each of these you would get 2 months or 4 months extra. That means it's literally time saving, it's literally money saving, and it's worth it. Okay, let's get back to question number 1. Question number 1 says match column 1 with column 2. In column 1 there is whooping cough, athlete's foot, German measles, cerebral malaria, and in column 2 there is bacteria, virus, fungus, and protozoa. These type of questions are very common in IIT JAM. So if you have guessed the answer correctly, it would be option C. Question number 2 is a time killer because it requires you to read all of these options and then answer. Which of the statement is false? The cell wall of archaebacteria is made up of pseudopeptidoglycans. The membrane lipids of archaebacteria are ether-linked, branched aliphatic chains containing D-glycerol phosphates. According to the structure of cell wall, archaebacteria are more similar to gram-positive bacteria. Archaebacteria are always pathogenic to human. Correct answer is the last one. This statement is false because archaebacteria are generally not pathogenic and they are kind of harmless. Then question 3. Cyanobacteria are photolithoautotrophs, which means they derive energy from chemical substances. They use inorganic compounds as electron donor. They use organic compounds as carbon source. They are commensal to animals. Here the correct answer would be option B. Whenever you hear the term litho in these particular complex nomenclature, you should know that they derive their electrons from inorganic donors. So this is kind of like a bacterial nomenclature kind of question based on nutritional requirements. So for this particular question type, you would get a nice explanation in the I button. So click on the I button to get it. And in that particular video, I have really broken it down for you guys to understand. Okay, match the antibiotic with the inhibitory mode of action. So you have a bunch of antibiotics in the left side and their mode of action on the right hand side. This is another type of question which is really common. So you should have a list of antibiotics and their mode of action in your fingertips. So they talk about penicillin, rifampicin, tunicamycin, sulfamide. So all of these names are really common. Names such as like puromycin, tetracycline can also be in this list. So you should have a list ready for yourself. But anyway, if you have answered properly this question, the correct answer would be option D. Here, notice that most uncommon antibiotic that I talked about is basically tunicamycin. Tunicamycin actually perturbs the glycosylation, especially the N-linked glycosylation. So it works very differently than the other antibiotics. Then question number 5. Which of the statement is false? Enriched culture media refers to the media which contains the nutrient that can support the growth of fastidious organisms. Selective media inhibit the growth of unwanted microorganisms and support the growth of organism of interest. Minimal media contains minimum nutrients possible for fastidious bacterial colony growth. Chromogenic media contain a combination of selective ingredients and chromogenic substances that, when incubated with target bacteria, produce a distinct color. So this is basically from bacterial culture media. If you want a quick revision on that, you can click on the I button to get to know about it. Here the correct answer would be option C. Minimal media would allow non-fastidious organisms to grow, but fastidious organism has complicated growth requirement, so a minimal media cannot support their growth. Which of the column is wrongly matched to the disease stated? So there is bubonic plague, there is tuberculosis, malaria, and ringworm. So you have to read all of these options, and if you have guessed it correctly, the wrong answer here is actually option B, but incorrectly stated here. The option B is the wrong answer here. I mean the correct answer here. So Microbacterium tuberculosis caused tuberculosis, not Microbacterium leprae. So sorry about this particular technical mistake. All of the following are examples of RNA viruses except, so this is again a very common type given that right now there is Omicron and coronaviruses. So SARS-CoV-2, influenza virus, Zika virus, adenovirus. So which one of that is not an RNA virus is the question. So adenovirus is actually a double-stranded DNA virus. A bacterium that has no defined shape and are not sensitive to vancomycin is Mycobacterium, Mycoplasma, Bacillus, Yersinia. So here the correct answer would be Mycobacterium. So Mycoplasma. Mycoplasma is one of the bacteria which are smallest possible bacteria known ever, and Mycoplasma is insensitive to any kind of beta-lactam antibiotics such as penicillin, vancomycin, etc., which acts on cell wall because it doesn't have any cell wall. So if you want to learn more about Mycoplasma, click on the I button. By the way, in I button you would get a list of microbiology topics which are most important and that you should know before you appear for IIT JAM. So it's a quick revision as well. The playlist is itself a quick revision. A pathogenic bacteria X can be differentiated based on gram staining, can't be differentiated based on gram staining, but it takes up acid-fast staining and containing high levels of mycolic acid in the cell wall. Most likely this bacteria is Yersinia pestis, Mycobacterium tuberculosis, E. coli, Corynebacterium diphtheria. The correct answer is Mycobacterium. So the genus Mycobacterium has very high level of mycolic acid in their cell wall and they don't take up gram stain, but they do take up acid-fast stain. Then the last question is from actually a IIT JAM paper, probably 2013 or 14. It talks about these kind of mode of nutrition like photoautotrophs, photoheterotrophs, chemoautotrophs, chemoheterotrophs, all of these things, which pretty much looks complicated, but I would strongly suggest before solving this question, right now, click on the I button, go to the bacterial nutrition, come back and then solve the question. It would be damn easy for you, okay? So let me know the answer in the comment and it would be really worth your time. Check out that microbiology playlist. Don't forget to like, share and subscribe. Thank you.